Every single day, your body is doing something, well, something more complex and precise than any factory on the entire planet. It's copying its own blueprint, not just once, but trillions of times over. So let's get right into it. We're going to dive into this absolute masterclass in molecular engineering, the incredible process of DNA replication.
Okay, so here's how we're going to tackle this. We'll start with the blueprint itself. What exactly is it? Then we'll see how the cell literally unzips it to read the code. After that, we'll meet the construction crew, this amazing replication machine. We'll also look at how it aims for perfection, because mistakes are a huge deal. And finally, we'll connect all the dots and see why this tiny process has massive implications for your health.
So, how does your body pull that off, making trillions of new cells every single day? Well, the answer, it really gets to the core of what makes us us. It all boils down to one fundamental process: copying life's instruction manual and doing it almost perfectly. That's how one cell becomes two, then four, and on, until you have, well, you.
Right, so before we can even talk about making a copy, we have to understand the original, right? Let's take a look at DNA's job as the cell's ultimate instruction manual. There's this fundamental rule in all of biology called the central dogma. It's basically the flow of information. DNA gets transcribed into RNA, which then gets translated into a protein. Simple enough, but hold on a second. Before you can do any of that, before you can build a single thing, you need a copy of the blueprints, right? That's where replication comes in. It's step 0, the absolute first, most critical thing that has to happen. Okay, so the cell's got the blueprint. What's next? Well, think about it like this. You can't read a book if it's closed, right? The cell has the exact same problem with its DNA, and the way it solves this is just beautiful, incredibly elegant. And here is the first major hurdle, that famous double helix. I mean, it's an amazing structure for keeping information safe and sound, but it's also a major physical barrier. Those two strands are wound up tight, held together by these hydrogen bonds, kind of like the rungs on a twisted ladder. The genetic code is literally locked inside. So how in the world does the cell get it open? It brings in this amazing little enzyme called helicase, and its job is simple. It's a molecular zipper, but it works in reverse. It just latches on, starts cruising down the DNA, and literally unwinds the helix, breaking those bonds and prying the two strands apart as it goes. And as helicase just chugs along the DNA, it creates this Y-shaped opening, and scientists call that the replication fork. And you can think of this fork as the active construction site. This is where all the magic is about to happen. All right, the blueprint is open. Time to bring in the construction crew. And listen, this isn't just one or two workers showing up. We're talking about a massive, highly coordinated team of proteins. The whole shebang is called the replisome. But right away, the crew runs into a huge problem. See, the two strands of DNA are anti-parallel. That just means they run in opposite directions, kind of like lanes on a highway. The big issue? The main copying enzyme, this thing called DNA polymerase, is a one-way street kind of worker. It can only build in one direction. So what do? This creates this really clever but weird situation. One strand, the leading strand, gets copied smoothly, all in one go. But the other one, the lagging strand, has to be built backwards in a bunch of short little pieces. And these short little pieces on that lagging strand, they have a name: Okazaki fragments, named for the husband and wife team who figured this out. It's just such a brilliant solution to that directionality problem, isn't it? If you can't build one continuous road, well, you just build it in small sections and then stitch them all together. So how does that stitching actually work? It's like a perfectly choreographed 4-step dance. First, an enzyme called primase comes in and puts down a tiny little landing strip, an RNA primer, to show the polymerase where to start. Second, DNA polymerase lands and builds that short Okazaki fragment. Third, a cleanup crew comes in and removes the primer from the previous fragment. And finally, an enzyme called DNA ligase comes in. It's literally molecular glue, and it seals the gap, joining everything into one beautiful, continuous strand. Okay, so building the new strands is impressive for sure, but building them perfectly? That's the real challenge. And the cell is absolutely obsessed with quality control. I mean, think about it. A single typo in the billions of letters of your genetic code could be, well, catastrophic. This is what scientists call the fidelity challenge. When a cell divides, it has to pass on a nearly perfect copy of its DNA to its daughter cells. Any mistake, what we call a mutation, can cause huge problems down the line. So, to prevent this, the cell has developed some truly incredible safety nets. So, how does it pull off this level of perfection? It basically has a three-tiered security system. First up, proofreading. The DNA polymerase enzyme itself has a delete key. If it puts in the wrong letter, it can feel it, back up, cut it out, and put the right one in. It's incredible. Second, you've got these checkpoint proteins that are like security guards constantly patrolling the DNA, looking for any damage or errors. And third, if they do find a big problem, they can sound the alarm and hit the emergency brake, stopping the whole cell division process. This gives the cell time to make repairs before passing on a mistake. So, okay, we've seen this amazing molecular machine in action. But why does any of this really matter to you and me? Well, it's because understanding this process is absolutely critical. It's directly connected to our health, to diseases, and to the very idea of what we pass on to the next generation. And that brings us to a really important question. This is where the tiny, microscopic world of the cell smashes right into the real world of medicine. Because when this incredibly fine-tuned, almost perfect machine breaks down, the consequences can be truly devastating. When you get errors in the code, it's a disaster. If those proofreading and repair systems fail, mistakes, mutations, start to build up in the DNA. This is the root cause of many genetic diseases, and it's also one of the fundamental things that drives cancer. Actually, that's how many chemotherapy drugs work. They're designed to attack and shut down this exact replication machinery, specifically in those super fast-growing cancer cells. All right, so let's wrap this up. If you remember three things, make it these. Number one, DNA replication is semi-conservative. It's a fancy word that just means every new DNA double helix is a hybrid. One old strand, one brand new strand. Number two, the whole job is done by the replisome, that all-star team of enzymes working in perfect harmony. And number three, the process has ridiculously good quality control, with proofreading and checkpoints to make sure the copy is as close to perfect as cellularly possible. And here's a final thought to leave you with. This isn't just some textbook science lesson. Right now, as you're sitting here listening to my voice, this incredibly complex, high-stakes machine is running in trillions of your cells. It is flawlessly copying your personal blueprint for life. It really makes you stop and think, doesn't it? What other amazing biological marvels are going on inside of us every single second that we just completely take for granted?